Good afternoon. My name is Dr. Antoinette Cordova, and today I'll be speaking to you about high-resolution 3D fluorescence imaging. I will focus on the methodology, the workflow, and the preclinical applications. So today I will be going over CFT or Cryo-Fluorescence Tomography. I will also be highlighting our technology, Xerra, our imaging platform, the research advantages of using CFT. I will also highlight the workflow, in addition to the preclinical applications that have spanned CFT technology. So let's begin. What is cryofluorescence tomography? It's an imaging technique that captures 2D fluorescent and white light images of ex vivo serial sections and compiles them into 3D images. On the left-hand side, you'll see a cynomolgus head, in addition to a whole mouse model, a rat brain, and a mouse kidney. So there is a volumetric range that can be interrogated using CFT technology. So why CFT? CFT actually fills the void between cellular microscopy and in vivo imaging modalities like MRI, CT, SPECT, PET. It also allows for orders of magnitude of interrogation and also resolution that is not accomplished between either microscopy or in vivo imaging. Now let's meet Xerra. Xerra is our CFT imaging platform, which features a 12 megapixel high resolution camera, a 10 micron slicer, 6 high-powered lasers ranging from 470 to 780 nanometers, in addition to 7 narrowband emission filters ranging from 500 to 850 nanometers. The Xerra is quantitative. It also offers micro resolution. So you can actually come up to the Xerra and you see this touchscreen here. A researcher can set up their study design and automate the process with ease of use. Once the researcher is finished loading the block, putting in their study design, you can walk away and use different desktop software to check in on your study or wait till the next day when it's complete. And ultimately, the Xerra allows for 3D fluorescence imaging. Now let's look at the research advantages of using the Xerra and CFT technology. The first is superior resolution. It provides resolution down to 20 microns for detailed anatomy. It also is highly sensitive. You can detect signal deep in the tissue. For example, it overcomes some of the limitations found in vivo modalities like IVUS, due to the fact that we are slicing slice by slice through using this tomographic technique. It's also high throughput. You can process several mice or multiple dissected organs on the several blocks that we offer. In addition to being robust, CFT will combine or integrate into your imaging workflow, whether you do in vivo imaging or tissue microscopy or both. You can also co-register your CFT data with other modalities using our proprietary software called vivoQuant. So two more to go, streamline. What's really great about CFT technology is it does not require any fixation or perfusion of the tissue, nor tissue clearing or radio labeling. So you can actually maintain complete anatomical context of your model when imaging. And lastly, it's versatile. It allows for visualization from 470 to 780 nanometers. Now I will highlight the Xerra workflow. The experimental setup is as follows. You determine your field of view. Currently, we offer 5 different fields of view with different resolutions for each. You then put your animal model in OCT and freeze the block, as can be seen here. Once the tissue is embedded in the block is then mounted into the system. And once you have done and gone through the study design process in the automated touchscreen, you're able to start your study. Once you do so, the camera images the block in fluorescence and white light. Once it has done that for each slice, a tissue is then sliced again and imaged again, and this process is repeated hundreds of times. In the end, you'll have an image stack that is error corrected and reconstructed into a 3D model that can be further analyzed. So basically, from beginning to end, you will be able to see what you could not see before. You go from here, the left-hand side, your animal model embedded in OCT, to a high resolution scan, where you can interrogate up to 10 microns of tissue removed per slice. You repeat this process 100 to 1,000 times, depending on the size of your animal model, and you will obtain a 3D aggregate in addition to 2D white light and fluorescent images. So Xerra and CFT technology will help you replace months of tedious lab work, and it will also reduce the use of animals because you will be able to get one additional data point and additional data you didn't traditionally get in your animal studies. So to highlight one more time, the exponential volume of data you get focuses on, first, comprehensive biodistribution, those interested in drug discovery, total tumor burden, those interested in oncology models. It also allows for multiplexing, the using of different fluorophores. If you're going to highlight, for example, cells labeled with, let's say, GFP, and you would like to use a different fluorophore to determine biodistribution of the drug, you can use that and do that using CFT technology. As I mentioned before, you maximize the data you get from one animal, and you also get complete data sets. You will get a complete data set of fluorescence and white light images in the 2D, and you will get your 3D reconstructed images. Now I will highlight the applications. So for CFT technology, there's a whole host of different applications that range from gene expression to neuroscience, to immunology, to nanotechnology. So let's begin. Before I do so, I wanted to just compare CFT technology to traditional in vivo imaging, which is like IVIS, for example. So on the right-hand side, you see the in vivo fluorescence images of these ovarian cancer cells that were labeled with TD tomato. And then there's the HER2-specific protein that was labeled with an Alexa fluorophore 647. When those animals were then put on the Xerra and CFT images were acquired, you can see the depth of information really just adds great value and honestly highlights what you could not see in traditional in vivo imaging. Now I will speak about gene expression. So why gene expression? It's very important for those that study these models to understand the role of genes and how their expression on behavior and disease phenotypes affects their animal models and also disease paradigms. So let's highlight that. So in this study, we're looking at bioluminescence versus CFT. So in this 4T1 mouse mammary tumor cell line, it actually expressed both luciferase and DS-red. So here we see the bioluminescence image on the left-hand panel. And what we did notice when we did run these animals using our CFT, or I'll put them on the Xerra, that this metastatic tumor model really missed a lot that the CFT actually ended up showing in this 3D image. We show that metastasis is in the abdomen, the lung, and bone, and this was not previously detected on the bioluminescence image. Now let's highlight drug discovery. As you know, drug discovery is very important and is a valuable tool for characterizing a drug candidate. Mainly, we look at 3D biodistribution, the localization of small molecules, antibody drug conjugates, in an effort to really elucidate the pharmacodynamic data, its affinity and selectivity for a target, and its stability in an animal model. In this study, we're looking at the intrathecal injection of SD rats with an AAV9. The AAV9 was labeled with an iodine 125, and it also expressed RFP. So initially, this was imaged using SPECT-CT at the 8-hour mark, and you're able to see the radio label on this left-hand panel. When these animals were then imaged using CFT technology looking for the RFP, we were able to see that the drug actually escaped the spine outside in 4 weeks. This would not have been previously determined if not actually using CFT technology and actually putting this model on the Xerra. Another highlight to drug discovery is this study with the dual tracer kidney imaging. So in this experiment, the researchers actually were looking at the drug development of an intrathecal drug. So they were evaluating 2 different routes of administration. Tracer A, which was intrathecally injected at time 0, and Tracer B, which was then done 15 minutes later. As you can see from the images, they were able to highlight the differences between the intrathecally injected drug and the one obviously given via IV. But what I wanted to highlight here is the vasculature of the kidney and how it can be highlighted. And that is really due to the fact that we also collect high resolution white light imaging. This actually provides the anatomical context of this vasculature in the small kidneys. Now I will go on to move on to immunology. And obviously the importance of immunology is that we can really look at organ-specific degenerative diseases and just the engagement, the mechanisms of action of immune reactions in vivo models. So in this multimodality macrophage imaging, we looked at 3 different types of imaging. So these mice were bearing xenografts and they were actually administered V-Sense. V-Sense is a perfluorocarbon nanoemulsion, and it contains fluorine-19 and an NIR fluorophore. So let's start with the first image. The first image is from IVIS, and as you can see, it's a low resolution image. When compared to then F19 MRI, we then struggle with the limitation of low sensitivity. But when these animals were imaged using CFT or the Xerra, we were able to look at the animal completely in a high resolution and highly sensitive manner and understand the macrophage tracking and the differences between the different modalities. Now let's look at cancer biology. So CFT can be very useful when you study tumor models, including different microenvironments, tumor heterogeneity, metastatic spread, and also looking at specific biomarkers. I think CFT, in addition to the fluorescence aspect, also adds to the value with the white light imaging because you can anatomically landmark tumor localization and also evaluate tumor metabolism. So in this study, the mice, the macrophage study, these mice again were bearing the xenografts and were given V-Sense. And in this model, we were able to look at the tumor heterogeneity that's highlighted using the V-Sense, as opposed to the MC38 cells. And we were able to evaluate the differences and such. And I also wanted to highlight that we can also look at the anatomical context of this using the white light images on the left-hand panel. And now I will highlight nanotechnology. Nanotechnology obviously allows for cells to be targeted specifically, and it also enables detailed imaging of tissues. And nanomaterials with a fluorescence group allow for visualization in CFT. Again, just highlighting the same study with the macrophage tracking using multimodality approach, using the nanoemulsion of V-Sense, we were able to visualize the differences in this model, especially in tumor heterogeneity. Now, in regards to neuroscience, it's very important, obviously, to focus on neuroscience because understanding the anatomy, the disease etiology, and the progression of disease using fluorescence visualization can be accomplished using CFT, as the next study I will show. Here we're looking at PK in the brain. So in this study, we looked at the delivery of antisense oligonucleotides that were labeled with a size 7, and they were then evaluated in mouse brains. There were 6 different time points ranging from 1 hour to 12 hours. And as you can see in the images here, the differences in the biodistribution from 1 hour to 12 hours is actually seen in these images. One other point that I wanted to highlight, in addition to understanding the biodistribution in a time-dependent manner, which is obviously very important for PK and PD, is that in addition to that, we were able to co-register the white light data with a brain atlas, and we were able to then look at where the conjugate actually localized to what areas of the brain. And you're able to do that using our proprietary software, VivaQuants. And in this case, they were able to map out, okay, at what different time points was the conjugated therapeutic localized, for example, in the cortex or the hypothalamus or the midbrain. And lastly, I just wanted to focus on how CFT complements histology. So in this mouse brain, we were able to actually acquire a slice during the CFT process. So for example, when you start a study, you're able to keep track of like the progress by actually looking either directly at the screen on the Xerra, or you can use any desktop platform to monitor. Once you get to your area of interest and you want to collect a slice for a standard histopathology workflow, you're able to do that. You're able to pause the Xerra, put it in slice collection mode, and then once you collect your slice, you can then perform standard histology and then image, for example, on a confocal microscope, or compare to any other histopathology workflow you may have, as seen here. So in conclusion, CFT is a high-resolution imaging modality, as shown and evidenced by the images I showed you today, ranging all the different preclinical applications. In addition, it builds upon and complements your existing imaging workflows. And lastly, what's most important is you actually maximize the data you collect from one animal. So you will get an additional data set, and you can even design experiments for the endpoints of studies that you traditionally would not have had. So I thank you so much for your time. If you would like more information, please feel free to contact info@immitimaging.com, or you can visit our website at immitimaging.com. Thank you.